everyone. This is Zeshan Shoaib. I hope you are all having a happy and healthy life. Today I am going to share an article with you whose title is Concurrent Measurement of Cerebral Hemodynamics and EEG during Transcranial Direct Current Stimulation. This article is published by Martina Giovanilla and other researchers from Barcelona Institute of Science and Technology. This article is published in 2018 in Neurophotonics journal, whose impact factor is 3.593, and this is a Q1 journal in optics category. In this article, they used three different modalities to measure the neuromodulation caused by tDCS stimulation. The modalities they used are functional diffuse correlation spectroscopy and time-resolved FNAS and EEG. In this video, I will only talk about the results acquired by FNAS modality. This is an introductory slide about transcranial direct current stimulation. We know that tDCS is a portable wearable brain stimulation technique that delivers a low electric current to the scalp. A fixed current between 1 and 2 milliampere is typically applied in tDCS stimulation, and tDCS works by applying a positive or negative current via electrodes to an area of the brain. This is a neuromodulation technique that produces immediate and lasting changes in brain functionality. In today's presentation, I will try to find the answer of two important questions. The first one is that whether the shame stimulation can cause to increase the hemodynamic response or not, and the other question is that can the hemodynamic response increase after ending the real stimulation. So in reference to this article, we will try to find the answer of these questions, and this is the experimental paradigm they used in this study. The experimental paradigm consists of 5 phases: a 10-minute pre-stimulation duration, followed by a 10-minute stimulation period, and three 10-minute post-stimulation periods. In all the durations, we can see a red color which is representing 2 minutes of eye closed and the other 8 minutes for eyes open. And the total experiment lasted for 50 minutes. A 10-minute stimulation including 30 seconds ramp up and 30 seconds ramp down at the beginning and the end of the stimulation period. And this practice they used for anodal and also for cathodal stimulations. In the shame stimulation, the current was ramped up in up to 1 milliampere in 30 seconds and then brought back to 0 milliampere in 30 seconds. And after 8 minutes, it was again ramped up and down to stimulate the real stimulation sessions. The design gave the subject the same feeling as a real stimulation because the ramp up and down periods are usually the most noticeable moments. However, the stimulation was too short to not have significant durable effect on the brain activity. Here we can see the optode configuration they used in this study. They used a particular optode in which the tDCS electrode, FNAS source and detectors, and the optical probes were fixed simultaneously. Here we can see the EEG optode positions and all other modalities positions as well. The FNS source and detectors they used on the frontal part of the brain, and they used time resolved FNS system which works with three wavelengths 760 nanometer, 800 nanometer and 830 nanometer. A total of 20 participants they used in this study. All of them participated in anodal and cathodal stimulation experiments, but for shame stimulation, they did only 12 experiments. For the data analysis, in this table, we can see the number of subjects they used for each condition. Some of the subjects they excluded from the data analysis after applying some particular exclusion criteria. Here we can see that for shame stimulation, they used 12 subjects data for data analysis. For anodal stimulation, they used 18 subjects data for data analysis, and for cathodal stimulation, they used 17 subjects data for data analysis. For FNUS data analysis, they used three different techniques. The first of them is the differential path length factor, which is the most commonly used technique for data analysis for FNUS data analysis. And they also used fit analysis and variance analysis for the FNUS data analysis. And after that, for statistical analysis, they used linear mixed effects analysis to perform the statistical analysis, and they applied R package for this data analysis. Here in this slide, we can see the results for shame and anodal stimulation in the left frontal part of the brain. The black line is showing the stimulation duration. In these results, we will focus only on the DPF analysis results, which is red in color. So we know that DPF analysis is the most commonly used technique to analyze the FNS data. In this figure, we can see that the activation is almost same in both shame and anodal stimulation conditions. The authors discuss that the DPF analysis is sensitive with superficial effects. That is why it is showing a higher activation in even shame stimulation. This is the reason they used variance analysis and fit analysis in this article to show the hemodynamic changes due to tDCS stimulation. The other main thing we can focus is that in DPF analysis, the anodal activation having higher values even after the stimulation ends. So we can see that we can see the higher values of anodal stimulation in post stimulation duration. So here this is also the answer of the question that the values of oxyhemoglobin can increase even in post stimulation duration. The results obtained with the analysis of variance and by fitting are comparable and show smaller changes than the DPF analysis both in anodal and shame stimulation conditions. In this slide, we can see the grand average results of shame, anodal, and cathodal stimulations by variance analysis. So here in the upper figure, we can see that the oxy values are getting higher values in post stimulation duration even by variance analysis. In the lower figure, we can see the deoxy concentration changes as well, and we can see that the deoxy values are decreasing in anodal and cathodal stimulations, but not changing in shame stimulation. So we can say that the deoxy parameter can also be used to monitor the neuromodulation caused by tDCS stimulation. So in this article, we saw that by DPF analysis, the oxyhemoglobin concentration increases about 2 micromole in the left hemisphere. So this increase is seen in active and even in shame stimulation. In shame stimulation, where the brain is not actually stimulated, we can see the increase of oxyhemoglobin. So this may suggest that results in DPF analysis are contaminated by superficial effects present both during anodal and shame stimulations. The right frontal lobe of the brain does not present any changes in the oxygenation. And also we saw that the oxyhemoglobin does not recover during the 30-minute post stimulation duration. This is consistent with the fact that if the stimulation is applied for a period longer than 9 minutes, the effect of the stimulation persists long after the stimulation ends. So in the end, I will try to conclude today's presentation. Even I did not talk about FDCS and EEG in this presentation, but we can conclude that they have shown that FDCS and F-nurse are suitable tools to monitor cerebral hemodynamics during and after tDCS, and they can be used simultaneously with EEG, which monitors the neural activity. And using DPF analysis, the shame and anodal tDCS stimulations may have the same effects. And the other thing, we can conclude that the results obtained with F-nurse by variance analysis showed small changes of oxyhemoglobin and deoxyhemoglobin concentrations, but the effect of this stimulation was still detectable. And the effect of this stimulation may persist long after the stimulation ends. And in the end, we can say that the tDCS over the frontal cortex induced statistically significant changes across different stimulation sessions. In the end, I will say thank you to all of you for listening me, and I wish you a happy and healthy life